Good afternoon. Let's get started here. So welcome to the 2021 Agricultural Research Award presentation and seminar. So we appreciate you joining us either online or here in person especially. So for those online and those here at about 4 o'clock, as Thomas wraps up, takes a few questions, we'll transition to the tent out here in Ag Mall. And we've got a reception there, so we thought the beautiful weather would be great to take advantage of today and continue celebrating Thomas's success. So again, welcome and thanks. I'm Bernie Engel, Senior Associate Dean for Ag Research and Grad Education, and I'm delighted today that we're recognizing Thomas Hook. Thomas is a professor in Forestry Natural Resources and also is director. I started to write down Indiana-Illinois Sea Grant. I thought that would be the better order, but I think officially it's the Illinois-Indiana Sea Grant Program and does a great job with that as well. So Thomas is our recipient today. Just a couple of things, and I know he will talk about some of these in his talk. You've got additional information in the brochure. So Thomas has made truly outstanding contributions in applied fish ecology and aquatic ecology, and is focused really on the Great Lakes with many of those efforts. In addition, he's made significant contributions in our understanding of the effects of anthropogenic stressors, things like nutrient loadings, invasive species, climate change, fisheries harvest, on aquatic populations and communities. So the impacts of his work are greatly valued in the state, in the region, nationally, internationally. So Thomas, come on up. Let's get the award presented. So I think we've got a plaque. And Tom, I think you probably want a picture. So we'll move over here. I'm going to put the microphone down. So there might be a check that goes with that gets directly deposited, and some resources that support Thomas's program then. So Thomas, I think you've got about 40 minutes of presentation. We'll have time at the end for some questions. Those online, I think Renee's monitoring the chat. So as we get to the end, if you've got questions online, we'll be able to get those in front of Thomas. And we'll remind you later as well, if you're here and ask questions, please hold down the microphone so that we capture that for our colleagues online. The recording will also be available as well. So, Thomas, the floor is yours. And again, congratulations, and thanks for sharing some of your work today.
Cool. Thanks, Bernie. So I just want to start by saying thanks. It's a little bit weird to get an individual award when pretty much everything I do is collaborative. I've never written a single author paper. I think I've written two author papers. So more or less everything I do is in collaboration with folks. And I'm really pleased to get this award. I'm thankful for the folks that were involved in nomination, the people that wrote letters. And I'm really thankful for all the people that I've worked with over the years. I thought about listing all the students and postdocs, collaborators, etc., but it was going to get out of control. So pretty much everything I'm going to talk about today has involved grad students or undergrads in our lab or postdocs, collaborators here at Purdue and at other universities. I've also really benefited from working with really good colleagues here in FNR and also with Illinois-Indiana Sea Grant, good administrators. When we found out about this award, Bob Wagner sent an email around the department in the spring, and I got a number of congratulatory emails back. And I think the first one I got was from Marlene Mann, who I think many of you know passed away a few months ago. And I didn't collaborate with Marlene on research or anything, but she's just an example of somebody who supported the stuff that we do in our lab and supported the work that a lot of other people did. We benefited from funding by a bunch of different agencies. A lot of what we do is analyze data that other people collect And so I'm really thankful for the people that have collected those data, the agencies that have been involved in collecting those data. And I also want to thank my family that make what I do possible. My parents, my sister, my wife, Tracy, my daughter, Ella, who's using this as an excuse to skip out on going back to college after fall break. And then our son, Max, who's skipping this to go to soccer practice. All right, so I think about a month and a half ago, I was asked to come up with a title for this. And I came up with Supporting and Growing Great Lakes Research. And I kind of thought that was what I was going to talk about, but I hadn't really thought about what I was going to cover. And then as I reflected on it, I'm kind of going to talk about supporting and growing Great Lakes research. I'm basically going to kind of follow the normal playbook for this type of presentation. I'm going to sort of give a bit of an origin story of how I got into what I do, provide a research overview of the type of research we do in our lab. And I'm at the end going to talk about some opportunities that I've had to work on promoting and translating Great Lakes science. But to start with, I just wanted to say a little bit about what I do for those of you who don't know what we do in our group. So I kind of have two gigs. As a faculty, I teach, conduct research. Most of our research, as Bernie mentioned, is in applied fish ecology, looking at stressor impact in the Great Lakes. And then I also serve as a director of Illinois-Indiana Sea Grant. For those of you who may not be familiar with the Great Lakes, here's a list of some of the things that characterize the Great Lakes. This was a list that was put together by the NOAA Great Lakes Environmental Research Lab. I'm not going to read it to you, but the main point here is that the Great Lakes are really important in a number of different ways. And I particularly like the figure on the lower right, sort of demonstrating that the shoreline of the Great Lakes, this is just the US shoreline of the Great Lakes, is greater than the entire Atlantic shoreline. So it's a really large system, has a lot of benefits. I want to draw your attention to the bottom bullet here, because most of what we do is related to studying fisheries in the Great Lakes and studying the food webs that support those fisheries. This includes a $7 billion a year sport fishery, also commercial fisheries, and several fisheries that are of cultural importance to groups like Native Americans. In terms of how I got into the research that I do, I thought about sort of coming up with a narrative about how we grew up going to the Great Lakes and went fishing with my granddad growing up, but that would be BS because I didn't really grow up anywhere around here. I grew up in Sweden and Alabama. My family's a research family. My mom worked in a biochemistry lab. My dad was a professor of biochemistry, still is a professor of biochemistry, claims that he's going to retire. We'll see. And so a lot of the conversation around the dinner table at home was about biochemistry, which I kind of thought was dull and not very exciting at all. And so when I went to college, I had no intention of studying anything having to do with biology whatsoever. I went to Michigan thinking that I was going to study political science or international relations. I had a good grounding in biology. I had really good high school teachers, particularly in biology and math. And after a while at Michigan, I realized that I couldn't do political science. So I switched over to biology, thinking that I would do some sort of biochemistry, because that's what I was used to. But it never really spoke to me until I took a class my senior year in college that was a general ecology class. And I really got hooked into ecology at that point. And what really got me into it wasn't so much like the environmental ethic, the tree hugging. It was more the trying to describe natural systems with models and trying to wrap your head around these really complex systems using quantitative models. And I really enjoy that. And so after I finished college, I went out to Nevada, worked on a project studying birds for a year, which taught me that I didn't want to study birds. And I worked in an outdoor store for a bit. I worked at in a bakery. I met my wife. After a year, I thought it was time to go back to college or back to grad school. And I had no plan on going to Michigan, but that ended up being the best place for me. So I went back to Michigan. I worked with a guy named Paul Webb studying larval fish in coastal marshes in northern Lake Huron. So this is when I first got connected and started working on the Great Lakes. After I finished my master's, I had no intention of staying in Michigan. I was going to go elsewhere for my PhD. Tracy started grad school at Michigan. I ended up staying at Michigan for my PhD. as well, and worked with Ed Rutherford and studied recruitment dynamics of alewives in Lake Michigan, did a variety of different types of studies. One thing that was really cool at this time was that I worked at a co-op between the University of Michigan and the Michigan DNR, the Institute for Fisheries Research, which is shown in the lower right here. It's now been torn down, that building, and it's been relocated, and the whole institute has really shrunk, which is too bad, because it was a really valuable experience for a grad student to kind of get that perspective of what applied issues were that the DNR cared about, but at the same time be at a university. After I finished my PhD, I had absolutely no intention of staying in Michigan. But at that point in time, we had bought a house, we had a kid. So it didn't really make sense to leave. I had some opportunities to go to more of a traditional postdoc, but this weird position came up, a research investigator, which was kind of like a research scientist position, but they paid you less than a postdoc. So it was awesome. But it allowed me to do stuff like write grants and start advising students and things like that. It was really good training for a faculty position. And it was also at a co-op. It was at a co-op between the University of Michigan and NOAA, which again had really good benefits of sort of having 1 foot in an agency and 1 foot at a university. And I started to collaborate with a lot of people. One person that I started to collaborate with at that time on a project studying diaperia in the Great Lakes and the decline of diaperia was Marisol Sepulveda here at Purdue. And she mentioned that there was a position open here, encouraged me to apply. And then I applied for that position here and ended up interviewing, accepting the position, coming here in 2008. What really stands out to me from that interview was one of the people on the committee was Andrew DeWoody, and he told me that it seemed like I had a really good thing going in Michigan. He couldn't understand why I would have moved from Ann Arbor to West Lafayette, which I thought was a really, really weird recruitment strategy by Andrew. And I said, F him, I'm going to take the job anyway. So ended up coming here. I'm happy I did. And then since I've been here, I started working with Sea Grant in 2010 as the Associate Director of Research, and then started as the Director of the program in 2018. All right, what do we do? I don't know about other faculty here or people that conduct research. I find it really hard to sort of describe what my research focus is or my research foci. You know, we have to come up with lists for like websites or when we apply for jobs or when we go up for promotion, and we have to kind of group all of our stuff together. If I look back, sometimes I have like 5 research foci, sometimes I have 1, sometimes I have 2. They're all over the place. And so for today, I came up with 2 research foci. The first one being describing structure of fish populations and communities. And I've heard a lot of fisheries people say this, and I've said it myself, that counting fish or studying fish is a lot like counting trees, except for they're invisible and they move around. And I've always said that, but I didn't really know where it came from. And I was working on a book chapter a couple months ago, and I was like, I should really figure out where that quote came from. And so the actual quote here is by John Shepard from the University of Southampton. Managing fisheries is hard. It's like managing a forest in which the trees are invisible and keep moving around. And so kind of 2 points here. One is that foresters have it easy. I mean, it's just like a tree that sits in one place. It just doesn't move for 100 years, grows very slowly. I don't really understand what the challenges are. On the other hand, fisheries, is really hard. Like easy stuff, like what habitats do they use? When do they spawn? How fast do they grow? Really things that seem like they'd be pretty obvious are really difficult to describe for fish. The other research focus that we work on is trying to understand the effect of anthropogenic stressors on Great Lakes fish populations and communities. And so we try to understand things like effects of nutrient loading and climate change and invasive species on fish populations and communities. And in so doing, we use a variety of different research approaches. So we conduct field studies, we do lab studies, simulation modeling, meta-analyses. A lot of what we do is analyze large data sets that other people have collected. So we really try to figure out the right research approach for the question that we're asking, as opposed to having a really limited set of research approaches. So when I've seen these kind of talks before, when people start presenting research, they always kind of say that, I'm not going to show any research. I'm going to keep this really light. I'm going to not present actual data. And then they continue on and show like 30 slides of data. So I'm not going to lie. I'm going to show you some data, but I am going to try to keep it kind of high level. And I'm just going to go through sort of 4 example themes that we work on or have worked on in our lab, just to give you a feel. Try to give you a feel for the sort of research that we do. And so the first thing I want to talk about is ecological effects of hypoxia. So hypoxia is an issue in aquatic systems throughout the world. Here in the Great Lakes, there's a hypoxic zone that develops in Lake Erie, in the central basin of Lake Erie, every late summer, early fall. And that hypoxic zone has received a lot of attention over the years, and a lot of research has been directed at that. But there's actually hypoxia in a bunch of other places in the Great Lakes too. This is a paper that a grad student in our lab, Josh Tellier, who worked with Paris Collingsworth, myself, and others, documented that there's hypoxia in a bunch of other places in the Great Lakes. And so it's a much broader issue in the region than just central basin of Lake Erie. But even so, Lake Erie is the one that's received the most attention. And when it comes to ecological effects of hypoxia, there's really sort of 2 perspectives. One perspective is that it's catastrophic. These things are referred to as dead zones. You probably heard of the dead zone in the Gulf of Mexico or the dead zone in Lake Erie. So a lot of people are really worried about it. And there's probably reasons they should be. If there have been fish kills, there have been fish kills in Lake Erie because of hypoxia. There are fish kills down in the Gulf of Mexico because of low oxygen. And we know from our research that hypoxia affects distributions. Fish will get out of the way of low oxygen. They'll move up in the water column. They'll move horizontally. It affects what they eat. They can't reach some of their food. There's some evidence, even though it's not as strong, that it may affect how well they grow, their vulnerability to predators, their vulnerability to fisheries harvest. If fish are forced to stay in low oxygen, you can have all sorts of effects. This is from a meta-analysis that we conducted, where we went to the literature and looked at all sorts of studies where they have confined fish at different oxygen levels and then measured their consumption and growth. It was led by Alison Reisick and Zoe Almeida, who were both grad students in our lab. And what we found was that you could find a significant effect at about 4.5 milligrams per liter. That's a pretty high oxygen level. But consistently, oxygen levels got below 4.5 milligrams per liter, you could see an effect. So that's scary. That means that not only at really low oxygen levels are there an effect, but even at these pretty high levels, you can consistently detect effects on vital rates for fish. Anyway, that's one perspective on the ecological effects of hypoxia. Another perspective is that it's really not a big deal. It's a natural short-term phenomenon. In Lake Erie, it's around for about a month or so. And a lot of fish have adapted and can deal with it. So the image on the left, and the bottom left, is it's from Lake Erie, from a paper where we demonstrated that yellow perch will avoid the hypoxic zone. They'll swim up above the hypoxic zone, hang out above it, but then they'll dive down to the bottom and grab food off the bottom and come back up. So really, they're not that impacted in terms of what they're eating. We can show from experiments that we've done in the lab that a lot of fish, like yellow perch, can actually do quite well at pretty low oxygen levels. So there's some reasons to think that it's not that big of a deal. And then there's also an offsetting effect of fertilization. If you think about what brings about hypoxia, it's nutrient loading, right? So nutrient loading is problematic because it can lead to hypoxia and harmful algal blooms, but it also stimulates the growth of plankton and provides more food for organisms. And so this is a model that we developed to look at that trade-off, where we brought together water quality models for Lake Erie, foraging models for fish, and bioenergetics models to try to look at this trade-off between hypoxia negatively affecting habitat for fish, but then increased production positively affecting them. And the takeaway from this is that when it comes to something like yellow perch, it basically comes out as a wash. There's the positive effects of more food kind of compensates for the negative effects of low oxygen. But that's not the case for some other species. So again, most of what we work on is in the Great Lakes, but we do occasionally work in other systems. And A group of systems that we work in as well are natural lakes in northern Indiana. So there's about 500 glacial lakes in the northern part of the state. And Cisco are a native cold water species that used to be found in at least 45, probably more lakes in the state. As of the latest count, they're only still around in 6 or 7. 6 or 7 because depends on whether you think one lake is one lake or 2 lakes, depends on how you count it. And most of this decline is because they can't tolerate really warm temperatures and they can't tolerate really low oxygen. And so the high nutrient loading and the hypoxia that develops in all of these lakes has contributed greatly to their decline. So we've been doing studies to try to understand why have they been lost in the lakes that they have been lost, and now more recently we're trying to figure out how do they still persist in the lakes that they're still in. Everything points to those 6 or 7 lakes that they still remain in. The water is too warm, there's not enough oxygen, they really shouldn't be there. So how are they making it? All right, so shifting over to another theme now. So in Lake Erie, there's concerns about too much nutrient loading leading to hypoxia. In Lake Michigan, there's concerns about not enough nutrient loading. And that coupling that decreased nutrient loading with invasive mussels has led to an oligotrophication of Lake Michigan. So since the 1970s, the Clean Water Act was passed, the Great Lakes Water Quality Agreement came about. There's been a huge decrease in nutrient loading to the lake. Dreissenid mussels, first zebra mussels and then quagga mussels also have invaded the Great Lakes, and the filtering by these two invasive species is dramatic, and it's led to an overall decrease in production in Lake Michigan and overall spatial restructuring of the lake. And what I mean by spatial restructuring is a shift in terms of where production in the lake is occurring. So a lot of people have studied this. A lot of people have looked at what are the consequences, what's this new Lake Michigan food web look like using a variety of different methods. One thing that we did, and this was something that Margaret Hutton-Stadig did, working with myself and Paris Collingsworth and Barry Lesht. Margaret looked at satellite imagery, which we can use to measure chlorophyll concentrations, and looked at chlorophyll concentrations in different depth bins around Lake Michigan. So the red ones here are the depth bins that are really close to shore, and then the blue ones, purple ones, are further offshore. And what she documented was that the further offshore depth bins, we saw a huge decrease in chlorophyll concentrations over time, whereas the nearshore depth bins had actually increased. The mussels seem to be sequestering nutrients and production close to shore, leading to this sort of spatial restructuring where we have a nearshore shift in Lake Michigan, a nearshore shunt. And we've spent a fair bit of time trying to describe what that nearshore shunt means and that new food web in Lake Michigan means for the ecosystem using a variety of methods. A lot of what we've done is try to look at who eats who, and we use things like stomach contents, where we cut up their guts and look at what's in their guts, or fatty acid profiles, which is something that I've done reluctantly because it's kind of like biochemistry. And then stable isotope ratios, which is another way to trace who's eating who. And an example of what we found is captured in this figure, where a big collaborative project, the lead author was Ben Tershak, who was at Wisconsin-Milwaukee at the time. And we compared samples that we collected during 2010 to 2012 with historical samples from earlier, and showed that based on stable isotope ratios in the tissues of these organisms, there was a shift towards the nearshore. So delta 13 carbon is indicative of whether organisms are relying on nearshore production sources or offshore production sources. And overall, there was a shift towards the nearshore, which was consistent with this nutrient shunt, this production shunt that we were seeing. We've also looked at this using a variety of other approaches. We looked at diets of things like salmon and trout. When I mentioned this $7 billion sport fishery in Lake Michigan, these are the species that support that sport fishery. Most of them we found after this reorganization of the food web were still relying on alewife, which is an offshore prey that they've relied on historically. Their numbers are way down, but these organisms, these top predators, are still reliant on this species that's no longer very abundant in the lake. But some of them have switched over and are including other nearshore diet items. Things like brown trout, lake trout, and rainbow trout have shifted to include nearshore round gobies in their diets as well. Whereas some other species like Chinook and coho seem to almost entirely continue to rely on alewife. And another way of looking at that is looking at where the fish are being caught. So a lot of the sport fishery is done by charter fishing in Lake Michigan. And when the charter fishers go out, they have to report which 10-minute by 10-minute grid cell they go fish in. And they have to report where they fish, and they have to report how many people went fishing and what they caught. And we got a hold of all this data, which included over 500,000 fishing trips over about a 20-year period, and analyzed where they were catching different fish. And what we found is that fish like rainbow trout and lake trout, these species that were now relying more on nearshore resources, had switched over to feed on things like gobies and terrestrial invertebrates. Their catches had shifted closer to shore over time, whereas things like Chinook and coho, the ones that had continued to rely on the alewife, they hadn't really changed where they were being caught over time. All right, just to sort of talk about something almost completely different now, another area that we've worked on a lot is annual recruitment. So recruitment is basically population level reproductive success. It's how many young fish, like the one in somebody's hand here, are produced by the adult fish in a year. And it's highly variable across fish populations. So some years they'll have produced thousands and thousands of offspring successfully, and some it's almost zero. So it can be really boom or busty. And it really screws up management, because when managers try to plan fisheries and they have a lot of variability in terms of how many new individuals are coming into the population, it's hard for them to plan accordingly. So we try to understand that in a bunch of different ways. And this figure documents one of the studies that we did where we got a bunch of data sets on yellow perch that were collected by different management agencies from throughout the Great Lakes. And we looked at whether or not their recruitment was related to each other or correlated. And we found a pretty strong correlation. In other words, in a year when there was high recruitment of yellow perch in Lake Ontario, there tended to be high recruitment of yellow perch in Lake Huron and in Lake Michigan. So high recruitment synchrony, and it was related to climatic conditions. And we've done a bunch of different studies like this using a variety of data sets and a variety of different methods. What I thought I'd highlight today is a particular type of recruitment question that we've spent a lot of time on, which is looking at recruitment out of drowned river mouth lakes. These are shown in yellow here on the map, and they are small productive lakes that are connected to Lake Michigan. You can kind of think of them as estuaries. They're where rivers empty into the lake. We know from our work that they can be really favorable habitats for young fish. They're warm, they're productive, there's a lot of food. Young fish grow better here, they emerge earlier, they survive better. But at the same time, these habitats are also pretty small compared to all of Lake Michigan. So trying to understand how important these habitats, so part of what we want to do is understand how important these habitats actually are for these populations at the whole lake scale. And we want to understand how fish use these habitats. And one of the ways that we have done this is to use otolith analyses. Otoliths are ear stones that fish have. They're calcium carbonate structures that grow like crystals. And they're biologically inert in the sense that once material is deposited onto them, that material is not sequestered back into the body. And you can look at the very core part of an otolith and analyze the chemical makeup of the very core part of an otolith and get some clues as to where the fish spent early life. And so we've done that using oxygen-stable isotopes and carbon-stable isotopes. And we've documented that things like alewife that grow up in Lake Michigan, this is shown in blue here, they have a really distinct otolith core isotopic signature versus alewife that grow up in a place like Muskegon Lake, which is a drowned river mouth lake. However, when we look at who actually survives from these habitats, so this was after before winter, all the fish seem to move out of the drowned river mouth lakes. All the fish move into Lake Michigan. So the following spring in Lake Michigan, there should be a combination of fish that grew up in the drowned river mouth lakes and in Lake Michigan. And what we find, at least what we found in 2002, was that almost all those survivors came out of Lake Michigan. The drowned river mouth lakes basically seem to be unimportant when you scale it up to the whole lake, which is probably just because these systems are so small. We're going back and looking at this again because of the changes in Lake Michigan that have occurred. We think there's some reason to believe that things have changed and these drowned river mouth lakes have become even more important than they were before. It may have providing an even greater advantage for young fish that are growing up there versus in the main lake. We've also recently looked at similar questions for yellow perch. So this is a study where we used otolith chemical analysis of yellow perch, comparing them in drowned river mouth lakes versus in Lake Michigan. And when we did this study, we did a parallel genetic analysis too. And I'm showing the figure here, but you don't need to spend that much time trying to decipher the figure. I'm just going to show this with an animation instead. So in the summer, there's yellow perch that are in Lake Michigan, in the lower left shown here as whitefish, that were born in Lake Michigan. They're genetically Lake Michigan fish. And then there are fish that are in the drowned river mouth lakes. They were born in the drowned river mouth lakes. They're drowned river mouth lake fish. That makes sense. However, sometime in the fall and going into the winter, a good number of the Lake Michigan yellow perch move into the drowned river mouth lakes and hang out there over winter. And that's a little bit concerning from a management perspective because there's a lot of ice harvest that goes on here where people are fishing on the ice. And so a lot of the fish that are being harvested that are thought to be residents of the drowned river mouth lakes are actually Lake Michigan fish. And so we could be really underestimating harvest of yellow perch because it's actually taking place in the drowned river mouth lake. Then by the spring, they seem to be back out. They don't reproduce in the drowned river mouth lakes. They just go in there to overwinter. We don't really understand why they do that. And we don't understand why they don't just stay in the drowned river mouth lakes and reproduce because it's a more favorable environment for young fish than Lake Michigan. But that doesn't seem to be what's happening. Okay, the final sort of research theme that I wanted to touch on today was size-selective fisheries harvest. And this is something that a number of folks in fisheries are concerned about. It's basically the idea that the fish that get harvested by fisheries, be they recreational fisheries or commercial fisheries, tend to be the biggest ones. So we're removing the biggest fish out of fisheries. And people are concerned about that for a number of reasons. One reason they're concerned about it is because those large fish may be the ones that are producing the most fit offspring. There's some reason to think this because a lot of people have documented that as fish get older and bigger, they produce larger eggs. So the figure on the lower right here was a study by Zach Feiner and others in our lab where we collected walleye from throughout the Great Lakes region and collected their eggs at the same time and looked at the relationship between female size and size of their eggs. And there was a pretty good positive relationship. I'll point out that this is in diameter. So if you translate diameter to volume of a sphere, it ends up being quite a much larger difference. So bigger fish are producing bigger eggs. Smaller individuals are producing smaller and potentially less fit eggs. And we've seen this not only for walleye, but for other species in the Great Lakes as well. But does this really translate into better performance? So we looked at this. We looked at this on a few studies, and we haven't really found a relationship between female size and the performance of their offspring. Even though they're producing bigger eggs, their offspring aren't performing better. This is a study that we conducted at our aquaculture lab here outside of town, where we took yellow perch that we kept at the ponds, the aquaculture lab, and just as they were getting ready to spawn, we brought them indoors, we spawned them, we collected some of their eggs, measured how big they were, and then we fertilized eggs, let them hatch, and then we looked at how well the offspring performed, how well they survived, and how well they did. And what we found was that, as expected, larger females produced bigger eggs. However, larger females' offspring didn't perform better. As a matter of fact, they performed worse. And so at least in the case for yellow perch that we've looked at, there's not this evidence of a positive maternal effect where the offspring are doing better from large females. That doesn't mean that there's no reason to be concerned about size-selective fisheries harvest. There's a bunch of other things that can happen with size-selective fisheries harvest. And one of them is something called fisheries-induced evolution. Basically, when we're capturing the really big fish, we're creating an artificial selection pressure. And that artificial selection pressure can select for small fish, for fish that grow slowly, and it can select for small young spawners, where they spawn at a relatively small size in an early age. And we documented through some simulation models that it's very plausible that this could occur for Great Lakes fish. We did a review paper a few years ago that also argued for this. But I want to highlight some evidence of this for yellow perch. And here I want to look at yellow perch in Lake Michigan. And these are yellow perch from the Indiana waters of Lake Michigan. There was a huge commercial fishery for yellow perch in southern Lake Michigan back in the day, and so big that it crashed the population and led to the closure of the fishery. That's what's shown in the top right here. And over that time, there was monitoring that was conducted by the Indiana DNR and by Ball State. And we looked at the data they collected to look at patterns of maturation in perch. And what we found was that during the height of the fishery in the 1980s, fish had to were maturing at a pretty small size for a given age. So they were at age 3, age 4, age 5, age 6, once they reached about 160 millimeters, female perch were maturing. Once the fishery was closed and released, they shifted back and started maturing at a larger size for a given age. And so this is very much consistent with what one would expect to happen with fisheries-induced evolution, that once there's a release from that size-selective mortality, they evolve back in the opposite direction to mature at a larger size for a given age. What was surprising here is that it occurred so fast. And so our expectation was that this would be a much more gradual change. But what we saw for yellow perch in Lake Michigan was a very rapid shift indicative of fisheries-induced evolution. But in this, people haven't really documented this quick of a recovery in other systems. And it's kind of perplexing why we saw this for this population. All right, so I'm going to zoom out, and sorry for showing some data and dorking out a little bit, but I can't help myself. What I wanted to do now was kind of reflect on some opportunities I've had to be able to support Great Lakes research and help interpret Great Lakes research. And as I mentioned at the beginning, I didn't really have a great plan to get into working on the Great Lakes or working on aquatic systems. It kind of just developed over time. But through my career, I've really been exposed to a lot of things about the systems, and I've really come to appreciate the importance of supporting this type of research and for trying to interpret our understanding of these systems to a broader audience. And so I've been lucky to be able to do that in a bunch of different ways. And one way is through an organization called the International Association of Great Lakes Research. This is the scientific society that I most identify with, and we've hosted a conference here. I was on the board. I was president of the organization for a bit, and it's something I really value. I think it's a really cool organization because it's not just on a particular discipline. It brings all sorts of disciplines together, and it's focused on systems instead. So you get to talk to chemists and physicists and people that work in social sciences and managers. We also engage a lot with management agencies around the Great Lakes, things like state agencies like the Indiana DNR and the Michigan DNR. We work with tribal agencies. We work with federal agencies like NOAA, USGS, U.S. Fish and Wildlife Service. And I think this is really important because we want to make sure that the work that we do is communicated to those groups that can actually make use of that knowledge. It's a challenge to figure out how to best communicate science to management agencies, and it's something we're not perfect at it by any means, but having collaborations with management agencies and being in frequent contact with them, I think, is a good way to do it. And then obviously through education, through teaching classes, through working on textbooks and things like that, we're able to take some of our understanding and translate it to another generation. This is a picture of students from one of our FNR classes seining in Lake Michigan, where they're getting exposed to Great Lakes science hands-on. But far and away, the way that I've most come to appreciate the interpretation of Great Lakes science and how we can better manage the Great Lakes is through my work with Illinois-Indiana Sea Grant. And our mission is to combine research, extension, and education to empower Southern Lake Michigan communities to secure a healthy environment and economy. I can't talk about everything that Sea Grant does, but since this is a research talk, let me just highlight a little bit what we do relative to research. So I started as the Associate Director of Research just a couple of years after I came here, and Carolyn Foley started working with me at the same time, and she actually really ran the research program at that point in time. Then when I became the director, Carolyn became the research coordinator for Sea Grant, and then she really ran the research program. But what we've done over the years is we funded all sorts of different applied research. We fund research at multiple universities in Illinois and Indiana. We're affiliated with Purdue and with the University of Illinois, but we fund work at Notre Dame and University of Chicago, Loyola, all over the place. And I think kind of our niche is we fund applied research, and we're a lot more nimble than a lot of funding agencies. And as an example, if you think about invasive carp, like silver carp and bighead carp, when they were sort of first getting on people's radar, that's when Sea Grant was funding research on carp. At this point in time, federal agencies like the Army Corps of Engineers are funding research on the Army Corps, on invasive carp. And those federal agencies are not at all nimble. They're the opposite of nimble. So once they get around, something rises to the level that they can fund it, there's not really much that we can contribute at that point because they got way more money that they can fund, they can use to fund that work than we can. So our sort of niche has been to try to really focus on these emerging issues and try to put research funding towards things that seem like they're going to become a big issue in the future. Now, my role with Sea Grant now is really just kind of doing some stuff in the back and trying to set things up in a way that these folks can be successful in what they do. And what they do, the majority of folks in our program are involved with extension in all sorts of topics. I've listed a lot of the topics here. I'm not going to go through all of them, but some of the things that we've started to work on more here recently include aquaculture. So we've spending a lot of energy to inform people about aquaculture opportunities in the Midwest, inform consumers about where they might be able to get aquaculture products from. We do work with Great Lakes data. We've actively been trying to collect more data on the Great Lakes. Just a couple of months ago, we launched a buoy off of Navy Pier in downtown Chicago. So now we're collecting real-time data right off of downtown Chicago that's made available to the general public. We've grown in sustainable community planning and stormwater and green infrastructure so that we can help communities grow in a sustainable manner. And we've grown in education. And in particular, we've increased our capacity when it comes to K through 12 education. And when it comes to education in particular, but a lot of our programming, what we're trying to do is to prioritize working with communities that maybe haven't engaged with environmental organizations as much in the past. If you think about sort of traditionally who's benefited from environmental education or from environmental programs, it hasn't been the entire public. It's been certain groups. So I started off with the work that we've done has primarily supported a sport fishery that's worth $7 billion in the Great Lakes. But it's a pretty small fraction of people in the US that actually benefit from that sport fishery. There's a lot of people that's fairly unimportant to. And so we've really tried to develop programming that are inclusive and will benefit a broader audience around Illinois and Indiana. And I'm going to end here by just putting a little bit of a plug in for Sea Grant and pointing out that we fund a lot of research. We fund research for faculty, we fund research for grad students, fund programs for undergrads. And I'd encourage you all to visit our website and try to find out a little bit about what we do. And You can sign up for an email list there where you can get updates when there are research opportunities. We haven't actually had that many people from Purdue apply for our programs and for our funding, and we're hoping to spread the word so that more people will actually apply to Illinois Indiana Sea Grant from the College of Ag and from Purdue in general. And so with that, I just want to wrap up and again say thanks. I can't thank all the people that I've worked with over the years, but I found some recent pictures of grad students. This is before COVID, but some recent pictures of grad students and postdocs in our lab. And I want to thank all the people that have contributed to some of what I shared today. Thanks.
Thomas, looks like we've got time for questions. So, Jerry, looks like you're first up.
Okay. Thanks, Thomas. That was great. And I came specifically to get fishing tips, and
I'm not the person.
You actually provided quite a few, so thanks for that. Unlike you, I actually grew up going with my grandfather and my father and my uncles and my cousins and fishing not only the Great Lakes, but a lot of the inland lakes in Michigan and also a lot of those lakes on the northern side of Lake Huron. And in my family, the maintained assumption was that if you weren't catching fish where you were, that you should go somewhere else. So we moved around a lot and fished in lots of places, thinking that that was what was the underlying problem. But what I kind of understood through this process is that different lakes are managed differently. Some of them are not managed at all, and that there are challenges with these transboundary bodies of water that are managed differently by different entities on maybe either side of the lake. And so that strikes me as kind of a natural experiment. And not knowing much about fisheries research at all, I wonder whether those kinds of differences in management can be exploited or leveraged as research designs to learn more about how management affects fisheries as opposed to natural processes.
Probably. There are, if you look at the Great Lakes, there are a number of different entities that are involved in the management of the lakes. And it's not just, the US and Canada. That could be an issue, So in Lake Erie, the Canadians allocate almost all their walleye to commercial harvest. And in the US, they allocate the walleye for recreational harvest. They have very different ideas of how they're going to use a shared resource. But there's also tribal agencies that have their own rights. And then there are, in Lake Michigan, there are multiple states that border Lake Michigan, and they have different ideas. They have different ideas of what species should be stocked or what strains should be stocked, and they've had debates on what the right approach should be. So there's a lot of that sort of idea where there are different perspectives on how these fisheries should be managed. In terms of how you use it to set it up an experiment, it's a little tricky because there's no replication, so I'm sure you can gain insights from analyzing those different management systems and different social systems. But it's not obvious to me exactly how you would turn that into an experiment right off the bat.
Well, and conversely, to what extent does it become a confounding factor for you as a, fishery scientist?
Yeah. So, a lot of the management, as a fisheries researcher, I kind of feel like we provide recommendations and then people don't act on them or do act on them. So for instance, our data would suggest that the states in Lake Michigan may want to rethink whether they're going to stock Chinook and coho salmon, because it's unlikely that they're going to keep doing well in the lake because they're relying on this alewife food source that seems to be dwindling, and we should maybe start thinking about promoting these other species that are more adaptable. But that's not what they're doing because people want to catch Chinook salmon. And if they were to stop stocking or decrease stocking Chinook salmon too much, they get a lot of pushback from charter captains, from recreational anglers. And so it strikes me as not the most wise stocking practices. And it's a little frustrating, but it doesn't necessarily change the research that we do. So, I mean, one thing in the Great Lakes is that things are always changing. I mean, there's going to be new invasive species that are going to come in. We're going to have weird changes in nutrient loading. There's going to be new contaminants that come in. It's an interesting system because it's so messed up, and there's always new stressors coming in. So in some ways it's an opportunity as a researcher because there's always things to try to figure out. There's so much we don't know about the system still.
I have a really, really naive question, and it was right in the beginning when you talked about Lake Erie and the hypoxic zone. I did not know that it has the same hypoxic zone every year, and what causes it the same time every year? Do I have this right? And why is that? That's why I say it's a totally naive question.
Yeah, well, it's not exactly the same size every year, but it occurs in the same location. And it's in the central basin of Lake Erie. And Lake Erie has 3 basins. There's a western basin that's really shallow. There's an eastern basin that's really, really deep. And then there's a large central basin that's intermediate. And I've heard it described as the 3 bears from Goldilocks before, whereas the western basin is so shallow that it never stratifies. So if you don't have stratification, there's a limit to how much hypoxia you have. The eastern basin is so deep that the bottom water, where it's really cold, there's so much volume there that it can never run out of oxygen. And the central basin is just right. So stratification occurs a little bit off the bottom, and there's not that much volume there. And it's also really productive, so it can run out of oxygen. And the reason it develops is because of algal blooms that primarily occur in the spring and early summer, huge algal blooms, and then they grow up and then they die, and then they settle down to the bottom and they decompose. And because of stratification, that bottom layer is isolated from the top layer, so you can't replenish that. When that decomposition happens and oxygen gets used up, it's not replenished from the top. And so you get this hypoxia zone that develops. It's the same sort of idea that what you have in the Gulf of Mexico too. And it's some years it's bigger because of climatic conditions or how runoff occurs, how stratification occurs. Some years a little bit smaller, but it occurs in some effect every year. And the main nutrient contributor is the Maumee River, which drains a large chunk of Ohio. But according to some folks in Ohio, a lot of the nutrients that end up in the Maumee are actually coming from Indiana. So a lot of the I know folks here focus on Indiana's contribution to the dead zone in the Gulf of Mexico, but if you're looking at the northeast part of the state, there's a pretty big contributor to the dead zone in Lake Erie as well.
Okay, so after a whole career of research, award-winning research, here's my question. Is Lake Michigan too clean or not, thanks to the mussels?
Yes. If you're from a fisheries perspective, Lake Michigan is too clean. And maybe and clean is the wrong word, but it's we've done too good of a job of keeping nutrients out of Lake Michigan and the mussels have exacerbated that. So we just can't support the fish production in Lake Michigan that people want. And so there are some people that think that maybe we shouldn't quite do as good of a job of treating sewage. Like if we just half treated sewage and let it into Lake Michigan, that'd be a little bit better. Or maybe we should build pipes where we just pipe the sewage directly offshore into Lake Michigan. Obviously, that's unlikely to happen. But yeah, from a fisheries perspective, it's not productive enough anymore. And so it depends on what your priority is. Is your priority having really clear water, or is your priority having more fish production? And there's a bit of a trade-off there.
Can I follow that up?
Yep.
Is there any evidence of what sort of pre-European settlement fish populations would have looked like in Lake Michigan, and how current day compares to that?
Yeah, so that's a good question. So and even should that be our goal? I don't know. Yeah. So a lot of the species that are in here now are non-native, right? And the Great Lakes are kind of a Frankenstein system, and so yeah, there were native species would have been a lot of corygonids, which are like cisco, whitefish. There's other species of the same genus that were abundant. Near shore, there would have been things like yellow perch and certain types of shiners, which are minnows. I think I don't think we have a good idea of what the overall productivity was. We're probably closer now to what a natural system would be. So it depends on there's something called shifting baselines that people talk about in fisheries a lot, and I'm sure in other fields as well, which is this idea that you reflect back to something that you remember. And so people here reflect back to something when they were a kid 20, 30 years ago. It was a really productive system, but we hadn't really seen the effects of the Clean Water Act or the Great Lakes Water Quality Agreement. There was still a lot of runoff, but that's what people remember, and that's what they want. So we are probably, Lake Michigan limnologically now is getting close to Lake Superior. It's more similar to Lake Superior than it has when we've had a record of it. It's probably, there's not mussels in Lake Superior, so Lake Superior is naturally there's mussels in Lake Superior, but not nowhere close to the numbers that you have in the other Great Lakes. And this oligotrophication in Lake Michigan that's bringing it close to Lake Superior, the addition of the mussels may be making it more severe even than it would be naturally. But it's hard to know, and it's hard to know. And again, it's hard to compare what it should be because the species that should be there don't even exist. Some of them have been extirpated now, so it's hard to even think in those circles, I think.
I'm going to ask an aquaculture question.
Uh-oh.
So, like you said, NOAA has been putting a lot of money into aquaculture recently, and they are looking at offshore aquaculture, the Atlantic side, the Gulf side, and a little bit of the Pacific side. What do you think are the prospects for the Great Lakes, with the understanding that the Canadian side, they promote some aquaculture on their part of the lake?
Yeah. So, you know the story, but others don't, I don't think. So in the ocean, there's a lot of pen-based aquaculture. A lot of the salmon that we get are raised in pens in the ocean. We could do that in the Great Lakes. There's no aquaculture in the U.S. waters of the Great Lakes. There is I think just one facility still in Canada that's in Georgian Bay in Lake Huron where they grow rainbow trout in a pen-based aquaculture setting. But in the U.S., it hasn't happened. There was some discussion in Michigan where they started to explore this a few years ago, and people got pretty fired up about not having aquaculture in the Great Lakes. So there shouldn't be pen-based aquaculture in the Great Lakes. There's just not the support from it from the general public. I personally think it could make a lot of sense to grow it in the Great Lakes, and I think it's kind of weird because the same states that are stocking non-native fish into the Great Lakes, they don't want to grow those non-native fish in pens in the Great Lakes where they could be more readily harvested and sported. So I think I think one would have to do a lot of work to convince the general public that it makes sense, but I think it could make a lot of sense as long as you're growing fish that are the same fish that are already being stocked in the system anyway. But I think there's just there needs to be such a big change on public opinion about that before I think it could happen. So I think the amount of education that one would have to do on that would be enormous. I doubt it would happen anytime soon, even though I think it would make sense.
So as a follow-up to that, why is it that the general public is opposed?
I think they're worried because when it comes to pen-based aquaculture in particular, it's got a lot of negative publicity related to fish escaping, which I could argue, just to give the counterpoint, if it's rainbow trout that they're stocking anyway, what if they escape? You're already stocking them. It's an issue of they're worried about nutrient loading because a lot of times there's high nutrients that are coming out of those. Lake Michigan, Lake Huron is nutrient limited. Maybe it's not that bad of a deal. People are worried about escapement and then breeding with native population. There's no native rainbow trout in the Great Lakes anyway. And there's issues of disease. Another thing with pen-based aquaculture can also be that and if you're growing things like salmon and trout, is you have to feed them fish meal. It depends on the practices, but you actually, in some cases, they end up feeding them more wild-caught fish than they actually produce salmon and trout. So you're losing biomass in the process, right? It would be better for us just to eat the sardines or the anchovies that they're catching in trawls in the ocean, as opposed to turning them into salmon or trout. So that's another argument against it. But most of the argument doesn't make sense because it's about trying to prevent native or non-native aquaculture in systems where we're purposely putting in non-native fish. Yep.
So Thomas, I think we've got 2 questions online, and then maybe it'd be a great place to pause and we can move outdoors and continue some conversations. So the first one is, for the hypoxia condition, is there a way to increase the oxygen concentration artificially in the lake to prevent hypoxia? Is there any real application of that?
Yeah, so people do that in small lakes where they oxygenate the hypolimnion. They pump oxygen into the bottom, and another thing that they can do in ponds is they try to break down the stratification where they make it so that the lake or the pond is fully mixed. That wouldn't really help because we need that cold water. Oxygenation in Lake Erie, I've seen some people that have done calculations on this, like back in envelope calculations, and it's just too much water. Like the amount of energy in oxygen would be hugely problematic. I do think a really cool, and this may be something good for ABE students, a cool application would be, if you look at like the smaller lakes, like the lakes where the Cisco are in Indiana, where we're losing oxygen, those are much smaller systems. There's not as much volume. If we could come up with, if we're going to keep Cisco in Indiana, we're probably going to have to be pretty aggressive. And one way will involve oxygenating the bottom. And so coming up with some sort of clever way of being able to oxygenate the bottom of these lakes without using a ridiculous amount of energy would be great. But so far, people have tried it in larger systems. It just hasn't worked because the volume is of hypoxic water is just too great.
Thanks. The next question, do you have a favorite project that you've worked on, or one that for one reason or another sticks out in your mind?
One thing that I really, when I was going through my PhD and what I did for my PhD, I threw some code up there. And the reason I threw that code up there is because I was talking to a grad student in our lab the other day about that model, which is something that I developed when I was a PhD student, and he's working to adapt it now for something else. And I recalled so that paper that came out of that is one of the papers that I'm the most proud of. It was an individual-based model of alewife. It's been cited like 10 times. It's like, for some reason, it just never got the traction of other stuff. But it's one of the ones that I'm the most proud of. And the way that came about was in Ann Arbor there was a Pfizer lab, and you could volunteer for studies. We didn't volunteer; you got paid. You could sign up and get paid to do studies. And so I signed up. I guess we needed money. I was in grad school. Ella was like, I don't know, a year. So I signed up for this Pfizer trial, and I had to stay at this Pfizer facility for 4 nights or something, and take some malaria drug, I think, or an antibiotic. But the thing that was awesome was I wrote that whole model. Like, it would normally have taken me months, and for some reason, that was like the perfect situation. And I was more productive during that 3 or 4 day period than I was for like months ahead of time. Maybe it's because Ella was a baby and she was keeping me up all the time, and finally she wasn't around. I don't know. But that's the one that really stuck out, is one that I really, really, really enjoyed, was that really intense period of being able to develop this model and complete it in a really short amount of time.
Thanks. Let's give Thomas another round of applause.